Well, happy blustery day here in Florida. I'm Holly, and this is Botany with Holly, and we're going to be talking about photosynthesis and metabolic processes of plants, as well as chromatography, looking at the pigments that go into plants. So the concept of this lab is that plants obtain energy from photosynthesis. Okay, they get sunlight that comes in. The plant performs photosynthesis with light to power chemical reactions. That's the necessary thing to get the chlorophyll and the chloroplast to work is light. It creates like a little chemical powerhouse that yields energy and then eventually gets stored for food. This occurs in the leaves. Okay, the flower does not do any of the photosynthesizing. Chloroplast is needed as well as the stomas. So if you look on the underside of a leaf, they actually have like little mouths that go, like out, whatever. And this allows CO2 to enter and oxygen to enter and exit. This regulates also the water content because just as we transpirate, meaning or respirate, either way, transpiration happens basically through okay, and then respiration is the in and out process from what I understand. So the stomas actually let in the air as well as water content, and just as we let out water when we speak, or as they say, if you keep your mouth open all the time, that's how you get dehydrated. Carbon dioxide is needed for the plant to do its metabolic processes. As I said, it enters through the stoma, and then this goes through the plant to the chloroplast. Okay, the chloroplast is where all this happens. To convert the sunlight, the plants have to have pigment to absorb the light. So it's kind of genius. Why are plants green? Well, they're green because that's how it absorbs the light. So as they say, if you wear a dark shirt, black outside, you're going to absorb more light, whereas white reflects the light off, and that's why you don't get as hot with lighter colors. The darker the pigment, the more heat it absorbs, which makes you therefore hotter. Carotenoids, as in carrots, so this is like orange and yellows, are the primary pigments in plants. So green leaves reflect the yellow and green light off because they are already yellow and green. So it won't absorb that light, it bounces it off. Also because carotenoids, so if you look at color world, the interesting thing, and when I was a teacher, we used to say roigi biv, red, orange, yellow, green, blue, violet, or blue, indigo, violet. If you look at a color wheel and you kind of go across, those are all the complementary colors because they're opposites. They're complementary because they opposite one another and then they bring out things. Anyway, so yellow, green, violet reflected back, absorbs red, orange, blue, and indigo. Okay, so now not all plants have all the pigments, as I guess we're going to find out in this lab with spinach leaves. And that's where the chromatography comes in. Chromatography is the analytical method in which we basically separate and identify samples in a mixture. So color, chroma, as it were. What we're going to be doing is with paper chromatography. This is done with a chromatography strip. You can also do this, as I have done with students before, with a coffee filter. Coffee filters are great for pigmentation and doing all kinds of cute little art projects. And it will slowly separate out because it wicks it, right? If a leaf only has one pigment, we are only going to see one band of color. If it has multiple, it will show more. So now that I've explained what we're going to be doing, we're going to get started.